Our first session of conference is going to be one of our Into the Pipeline series. So as many of you who were with us last year may remember, we asked all of our industry partners to do a 6-minute talk test, a format we had borrowed from our friends at Action Duchenne. This gives us an opportunity to get a quick highlight of some of these clinical trials or approved products while still having time for you to ask questions to the companies. If you do want to learn more about any of these trials or products, we do have longer videos that are on the Conference Hub that you can dive into at your leisure. As a new addition this year, we have Dr. Mena Scavina, who has recently joined the PPMD staff, will be providing an introduction to these topic areas. So this first talk is going to be about different products that are aimed at restoring the production of dystrophin in the body. And so we'll start with that introduction video from Mena.
Dystrophinopathies, which include Duchenne and Becker muscular dystrophy, are caused by a variant or change in the DMD gene, resulting in little or no production of the dystrophin protein. These changes can include deletions, duplications, or nonsense mutations that disrupt the reading frame, which is the instruction the body follows to make a protein. When the reading frame is disrupted, it prevents the production of that protein, in this case, dystrophin. Let's look at the reading frame as a sentence. DNA provides the instructions for the protein, essentially like the letters in a sentence. Depending on which letters are missing or duplicated, the sentence may be completely changed and not make any sense. Or it may make sense, but not exactly like the original sentence. Therefore, the instruction to make the protein is altered, and either no dystrophin or reduced dystrophin is produced. This is seen with deletions, duplications, or smaller point mutations. If a period appears prematurely, then the sentence is shortened and may not make any sense. This is seen with a type of point mutation called a nonsense mutation, which inserts a stop signal into the gene. Stop codon read-throughs allow us to ignore the premature period, complete the entire sentence, and produce protein. If letters are missing, called deletions, exon skipping allows some letters that border the original deletion to be skipped over to correct the sentence and allow some protein to be produced. Antisense oligonucleotide, or ASOs, are single-stranded pieces of DNA that are used to restore the reading frame through exon skipping. If we can restore the reading frame or correct the sentence, we can produce dystrophin again, although it may be modified. The protein may be smaller and the amount may be less than typical.
And joining us tonight for this first panel, we have Diane Berry from Sarepta Therapeutics, Leslie Magnus from NS Pharma, and Christian Werner from PTC Therapeutics. As a reminder, as these discussions go on, if you have questions or comments, you can submit them to that box below the live stream, and we'll try to get to as many of those questions in the live Q&A as we can. And with that, we can begin with Sarepta. So Diane, whenever you're ready.
Thank you, Eric and Ryan. Can you hear me okay?
Yes.
Yes.
Great. Okay. Hello, everyone. I'm Diane Berry, and I am the Senior Vice President of Global Health Policy, Government, and Patient Affairs at Sarepta Therapeutics. I want to thank PPMD for inviting us to participate in this panel today. I'm pleased to give you an update on Sarepta's PMO exon skipping technology. I'm going to be relatively brief, so if you'd like to learn more, please view our pre-recorded PMO presentation in the Conferences On Demand library, or just reach out to us directly. Next slide, please. Please understand that some of our updates may be forward-looking, meaning I may speak about our future goals and the potential of our therapies, things that haven't happened yet. And with that, there is risk and uncertainty related to such statements. Next slide. Sarepta's experience in Duchenne runs deep, with over a decade of dedicated research and learning about the disease and what's important to patients, physicians, regulators, and payers. And in recent years, the breadth of our Duchenne pipeline has become unparalleled, spanning 3 distinct approaches. Our RNA platform, which includes our PMO and PPMO exon skipping technologies. This includes our 3 exon skipping products that are approved in the United States. We also have an active microdystrophin gene therapy clinical stage program and gene editing work in the early stages of development. If you're interested in learning more about our next generation PPMO or gene therapy development programs, we're addressing those in the live sessions over the course of this conference and also in videos in the on-demand library. Next slide. 3 of our PMO programs have been approved by the FDA in the US under accelerated approval. They aim to restore production of truncated dystrophin and slow disease progression. Ataluren is a PMO designed to skip exon 51. It was the first therapy approved in the US in 2016 to treat Duchenne and is intended to treat about 13% of the Duchenne population. Golodirsen in the middle here is a PMO designed to skip exon 53 and was approved in the US in December of 2019. And this PMO addresses another about 8% of individuals living with Duchenne. And casimersen is a PMO designed to skip exon 45. It was approved this past February, and we expect it to address another 8% of the Duchenne population. Together, these 3 PMOs are predicted to serve up to about 30% of the Duchenne community. Next slide, please. For patients and families who are interested in exploring access to our approved therapies, we have a team of case managers at the ready to help. Sarepta Assist is our patient support program that helps patients navigate the process of starting and staying on approved Sarepta therapies. Our case managers provide one-on-one personalized support to families to help with reimbursement support, financial assistance options, treatment logistics, and other treatment-related needs. You can reach out to us through the contact information listed here. Next slide. We are deeply committed to building a strong body of evidence for our PMO programs. We have 10 plus years working in Duchenne research, hundreds of study participants across all PMO studies, and 15 PMO clinical trials. Participants have ranged from 6 months to adulthood. In our job of data generation and understanding the long-term benefits and risks of therapy doesn't end at approval, we continue to study our therapies and collect data post-approval through both clinical trials and real-world evidence studies. For example, we've been able to collect up to 7 years of follow-up data on Ataluren-treated patients through real-world sources. We are thankful to the many participants, families, investigators, and site staff whose commitment and perseverance have made all this work possible as we continue to accumulate knowledge regarding the benefits and risks of our PMOs. We know we have a lot of work to do to develop treatment options for many more individuals with Duchenne and to advance the science and improve and build upon what's been accomplished to date. Next slide. I'd like to spend a moment sharing 2 ongoing late-stage PMO clinical trials. Essence is evaluating casimersen and golodirsen, and Mission is evaluating a high dose of Ataluren, higher than the FDA-approved dose. Both fall under the umbrella of what we call post-marketing studies, agreed to with the FDA as part of the accelerated approval process. The goal of Essence is to evaluate the efficacy of casimersen and golodirsen compared to placebo in patients with Duchenne. As part of this study, we're collecting functional data as both primary and secondary outcome measures. Essence is no longer recruiting in the US, but it's currently enrolling in other parts of the world. As for Mission, its goal is twofold. Part A is to evaluate the safety and tolerability of higher doses of Ataluren. We've enrolled this first cohort. And Part B is to select a high dose and compare its use with the 30 milligram per kilogram dose of Ataluren. In addition to collecting safety and expression data, we're also collecting functional outcomes in this study. Next slide. As for next steps, we remain committed to continuing to support all U.S. patients receiving our PMO therapies, executing our ongoing post-marketing studies, and further building the body of evidence in support of our PMOs. We are continuing to collect and analyze data to inform next steps for subsequent exon skipping trials. This includes data not only from our PMO platform, but also from our PPMO exon skipping platform, which we will be speaking to in a live session on Saturday, or you can view our video in the on-demand library. We will continue to work with regulators to determine a streamlined development and regulatory path for exon skipping therapies that target the more rare mutations. And we'll be sharing our future development plans with the community when we have these further mapped. And the last slide, please. I just want to thank PPMD again for having me here to speak today, and I look forward to being able to respond to your questions. And if you have any questions we don't get to or comments after this session, please reach out to our patient affairs team at advocacy@sarepta.com. Thank you.
Great. Diane, thank you so much, and thanks for being first out the gate. Never easy to do the presentation. I just want to do one follow-up question to some of your remarks. I know you mentioned you're collecting data in post-approval, so after patients get on drug to your approved products and sort of real-world evidence. I know PPMD is working to combine some of that data with you in our PRO registry. But can you talk a bit about what exactly you're looking at in real-world evidence and timelines for analysis on what's happening with these patients and really what may be challenges with collecting this type of data?
Sure. Yeah, no, happy to, Ryan. It's a great topic. There is so much attention these days and recognition, I think, on the importance of real-world data and real-world evidence. I think one thing you had mentioned, you had referred to specifically is our observational study that we're conducting through a registry on our approved PMO products at multiple centers in the U.S. This study in particular collects data that's collected in routine visits, so no special visits required or assessments in order to decrease the burden on patients and families. But this registry is really only one source of real-world data, really to help inform a better understanding of how our therapies work in the real world and capture the true experience of a patient as opposed to through controlled clinical trials. And what this means is really looking at data that tells how our treatment impacts patients' lives and well-being in a way that goes beyond just the traditional clinical measures. And this can be in the form of observational studies and registries, like I just mentioned, or electronic medical records, wearables, claims, and billing data. And we're exploring all of these data sources. We believe this type of data really is an important element to appreciating the overall comprehensive picture of benefits and risks that can supplement the clinical trials that we're doing, including post-marketing trials that we're required to conduct by FDA. And so our hope really is that real-world data and evidence can get to a point where it supports product approvals or can perhaps satisfy post-approval study requirements and certainly can help secure patient reimbursement for treatment. And so we have a lot of work going into this. And as I mentioned, a lot of attention. FDA's building a framework that Congress called for under 21st Century Cures. Just yesterday, Congress introduced Cures 2.0, or a discussion draft anyway, and again, reinforcing that there is a really important role in place for real-world evidence in regulatory decision-making. So a lot of forward momentum. I think the COVID pandemic really highlighted the benefits and potential use of real-world evidence and really required a shift toward more virtual data collection methods. And that's likely to lead to improved data collection platforms. I can't really speak to, you asked about timelines. I can't really speak to timelines right now. I think, we're still in the early stages, collecting some of this data and really understanding the capabilities of the various types of real-world data sources. And along with that, you asked about challenges. There are a number of challenges. I think some of the key ones really are center around data governance, how to preserve data security and patient privacy. Oftentimes there's incomplete data or there's variability in the measurement. And really, lastly, is really trying to figure out what is considered good quality data. And I'm sure you all have heard the adage, garbage in, garbage out. And so with real world data, it will only be good as the data source itself. So, we have to be really diligent to ensure the data is high quality and reliable in order to ensure it is usable.
Okay.
So, sorry, I hope I didn't take too long.
No, thank you so much. It's a big topic. We could potentially even circle back. So I'm going to turn now to our next speaker, Leslie Magnus from NS Pharma. Leslie, are you ready to go? Oh, you might be on mute. There you go.
Yeah, but not anymore.
Okay, floor is yours.
Please. Great. Well, thank you. And hi, Eric and Ryan. Nice to see you again after one very long and complicated year since our last virtual meeting. But again, thank you to PPMD for hosting this very important forum. And thank you for giving NS Pharma the opportunity to present some information about both the company and our compound Viltepso. If I could have the next slide, please, which is our forward-looking statement. So I'll leave that for just a minute. And then may I have the next slide, please? For those of you who may not be familiar with NS Pharma just yet, NS Pharma is the subsidiary of Nippon Shinyaku. We are based here in the US in Paramus, New Jersey. And our main area of focus is orphan rare diseases, specifically Duchenne muscular dystrophy. And with that in mind, we were the sponsors of the North American Phase 2 trial for Viltepso, and we are the sponsors of the global Phase 3 trial for Viltepso, also known as RACER 53. In addition to orphan rare diseases, we are beginning to broaden our platform with some work in the hematology oncology space. Our parent company, as I said, is Nippon Shinyaku. They're based in Kyoto, Japan. And they were founded in 1919, so they're over 100 years old. They have approximately 2,000 employees globally. And like NS Pharma, they focus on drugs for rare diseases and also hematology oncology. And in addition, they also focus on urological and gynecological products. Next slide, please. This slide represents some of the key milestones that have occurred since 2017. I won't touch on all of them, but I do want to point out some of the major highlights. Importantly, in October 2019, we started RACER 53, which, as I said, is the Phase 3 global trial for Viltepso. In December 2019, we completed the new drug application submission to the FDA, which led to the approval of Viltepso, now known as Viltepso, in August of 2020. So just under a year ago, this product was approved in the US. Presently, we have information on our long-term data, it's interim functional data, which will be presented today at this conference. Come the future, we anticipate initiation of another trial with Viltepso, this one looking at exon skipping in both ambulatory and non-ambulatory patients, and patients who are over the age of 8. And also in the near future, we anticipate initiation of another exon skipping type, exon 44 skipping, as I said, in the near future. Next slide, please. So just a bit more about Viltepso or Viltepso. It's an antisense oligonucleotide, which we heard about earlier in this call, and it's designed to skip exon 53. So therefore, it is effective in patients who are exon 53 skipping amenable, who have deletions that you can see listed here, 43 to 52, 45 to 52, even 52 alone can be treated with Viltepso. In addition to this list that you see on the slide, there are many other deletions that may be treated with Viltepso. And so you should always consult with your physician and or genetic counselor to find out if your deletion is amenable to exon 53 skipping specifically. In our initial phase 2 study, we looked at treatment with Viltepso at 80 milligrams per kilogram per week, and this resulted in significant increases in dystrophin production. The mean dystrophin production in the group of 8 patients was approximately 6% of normal once they had completed 20 to 24 weeks of treatment. In addition to looking at this biomarker assay, we also looked at timed function tests, and those provided supportive evidence of treatment-related clinical improvement. And those patients in our Viltepso group were compared to both age-matched and steroid treatment-matched natural history controls from the Synergy DNHS cohort of patients. At the conclusion of this initial phase 2 study, all participants were given the opportunity to enroll in a long-term extension study that would continue to assess both clinical and safety outcomes. All 16 patients elected to remain in the study, and some patients are even continuing at this point in time. Next slide, please. On this slide, we look at a more granular level at the results from the primary endpoint, which was the dystrophin production after 20 to 24 weeks of therapy. As you can see from this graph, at week 25, the change from baseline in mean percentage of normal dystrophin was statistically significant for Viltepso, with an increase of 5.3% to an average of 5.9% of normal dystrophin. Further, all patients exhibited an increase in dystrophin over their original baseline levels, and all but one patient achieved a dystrophin level of at least a minimum of 3% of dystrophin. When we looked at the safety profile for this compound, there were no serious adverse events, no treatment-related serious adverse events. There were no discontinuations from the study due to adverse events, and no deaths occurred during the study. What the safety profile did show were patients who had not unexpected symptomatology, such as nasopharyngitis, cough, nasal congestion, confusions, arthralgia, diarrhea, and vomiting, mild to moderate in almost in all cases. Next slide, please. On this slide, we're looking at the primary endpoint of the long-term extension study. This is data that's been collected at up to 109 weeks, so over 2 years of therapy. In this group, we see that the patients who received Viltepso, and we're looking at time to stand in seconds, so this is the primary endpoint. We see that in blue are the patients who received Viltepso therapy over this 2 year time period. In gold, we see the patients who make up the historical control group. What we're seeing with the blue line is a maintenance, a continuation of the same time to stand in seconds throughout the course of this study, while the historical control group shows an increase in time to stand. Keep in mind that on this graph, improvement is shown by a decrease in numbers. So if the graph is going up, the line is going up, that's a clinical deterioration. This data will be presented in great detail at our 8 p.m. session with Dr. Paula Clemens, and I hope everyone is able to participate and listen into that session. Next slide, please. So finally, I'd like to just review that Viltepso was approved in the US and in Japan, both in 2020. We're continuing to enroll patients in the phase 3 RACER 53 study. We do have an NS Patient Services Support Center. That number can be found on our viltepso.com website. That is to assist both patients and healthcare professionals with questions that they may have. We anticipate initiation of an ambulatory and importantly non-ambulatory exon 53 skipping study in the near future. And we also anticipate initiation of an exon 44 skipping study. So please, for both of these, if you have an interest, continue to check regularly on clinicaltrials.gov, where we update very frequently. Thank you. Next slide is.
Just one follow-up question to your slides. Similar question to you that I gave to Diane. Now that you have an approved therapy with Viltepso, how are you tracking those patients that are on that approved therapy to understand long-term outcomes, efficacy and safety ultimately? Is there a registry, or so what's the methods for that?
For the patients who are receiving commercial product, we do not have a registry at this moment. Of course, we passively receive spontaneous adverse events and collect those as per requirements of the FDA. So we would hear about those without a doubt, because these patients are being seen, even though they're not in a clinical trial, they're being seen every week at an infusion center at the time of their administration of the product. In addition, the patients that I've described from our 2-year data, many of them have gone on and are still in the study. So we'll have 3-year data when that completes. And now those patients are further being offered the opportunity to subsequently enroll in another long-term study where they will have periodic visits, and we will be collecting that data for 5 to 10 years of time from those same patients. So that will be a very rich data source of these patients who have been on Viltepso.
Okay, thank you very much. I'm sure we'll get into more discussion during the panel. We're going to switch now to Christian Warner from PTC. PTC Therapeutics has been in this community for many years, developing Ataluren and then bought Emflaza. Today, Christian's going to be talking about Ataluren, so I'm going to turn it now to Christian Warner.
Thank you very much, Eric and Ryan. Good afternoon, everybody. Thank you for the opportunity to talk to you today. My name is Christian Werner. I'm the DMD Therapeutic Area Lead working at Global Medical Affairs at PTC Therapeutics. And it's my pleasure to provide an update on Ataluren today, comparing data from Ataluren clinical studies with the Synergy Duchenne Natural History Study. Next slide, please. We are a public company, and this is PTC's forward-looking statement. Next slide, please. Ataluren is the first and only targeted medicinal product approved for the treatment of nonsense mutation Duchenne muscular dystrophy anywhere in the world. Currently, Ataluren is available in more than 40 countries worldwide, excluding the US, where it is an investigational product. Ataluren is indicated for the treatment of Duchenne muscular dystrophy resulting from a nonsense mutation in the dystrophin gene in ambulatory patients, age 2 years and older in the European member states, Iceland, Liechtenstein, Norway, Great Britain, Northern Ireland, Kazakhstan, Israel, and the Republic of Korea, and for patients 5 years and older in Chile, Brazil, and in the Ukraine. Next slide, please. PTC Therapeutics is conducting a post-approval registry called STRIDE to obtain information on the safety and effectiveness of Ataluren in routine clinical practice, where patients are followed up for 5 years. Data from the data cut in July 2018 showed 181 patients in the STRIDE registry that were statistically matched with 181 patients from the Synergy cohort who were comparable in established disease progression predictors. Ataluren plus standard of care in the registry was compared with standard of care alone in the Synergy Duchenne Natural History Study. Ataluren plus standard of care significantly delayed the median age at loss of ambulation by 3.5 years, which can be seen. The red graph is showing the patients treated with Ataluren in the STRIDE registry. The gray one are the patients from the Synergy cohort. Next slide, please. Looking at the non-ambulatory patients only, Ataluren-treated patients in the STRIDE registry showed a delayed lung function decline by around 3.1 years compared with matched Synergy patients, shown here for a data cut in January 2019. At this time point, 220 patients have been included in the STRIDE registry, coming from 58 sites in 14 countries worldwide. Next slide, please. Showing another study called Study 019. This was a phase 3 long-term multi-center international open-label study that included patients with nonsense mutation Duchenne muscular dystrophy who had received Ataluren in a prior PTC therapeutics sponsored study at investigational site outside the US. The primary objective was to assess the long-term safety and tolerability. In this study, patients were followed up for up to 240 weeks for efficacy assessments. This analysis showing a matching that was performed to identify synergy Duchenne natural history patients compared to study 019 patients, again, a comparable in established predictors of disease progression. Ataluren treated patients in this study showed a delayed loss of ambulation by around 2.2 years compared to the matched synergy patients. Next slide, please. Same as before, looking at the non-ambulatory patients only. Non-ambulatory patients treated with Ataluren in study 019 showed a delayed lung function decline by around 2.3 years compared to the matched synergy Duchenne natural history patients. Last slide, please. If we summarize the data that we have seen data from the STRIDE registry, an ongoing observational study evaluating the real-world safety and effectiveness of Ataluren in patients with nonsense mutation DMD. As highlighted before, as of January 2019, 220 patients from 58 sites in 14 countries have been included. The second study I've shown, study 019, was a phase 3 long-term multi-center international open-label study to assess the long-term safety and efficacy. Overall, Ataluren was generally well tolerated and had a safety profile which is consistent with what has been shown in previous clinical trials. The matched analysis presented today we used to evaluate the effectiveness of Ataluren plus standard of care in the STRIDE registry or respectively study 019 compared to the standard of care alone in the synergy cohort. Ataluren treated patients in STRIDE showed a delayed loss of ambulation by around 3.5 years compared to the synergy patients, and in study 019, a delay by around 2.2 years. Results also suggest that a trend toward delayed worsening of lung function for STRIDE and study 019 patients compared with those in the synergy Duchenne natural history study. For further information about these studies and our ongoing clinical trials, please have a look at the pre-recorded presentation on the platform. Thank you for your attention. I also want to extend our thanks to the boys and families for their participation in the studies and to everyone involved in the conduct of these studies. Thank you. Back to you, Ryan and Eric.
Thank you so much, Christian. That was great. It's really interesting to see the data. We know that this is a requirement in Europe with EMA. We're hopeful, and we'll talk a bit about the US side later in the panel. But one question I wanted to ask you was about sort of second generation drugs for nonsense mutations or read-through drugs. We're seeing in exon skipping the move towards the next generation, so how to make them more efficient, looking at peptide conjugates, et cetera. I know families in the US are wondering, with research in nonsense mutations, is there research for second generation, and is PTC looking at that type of research?
So, great question. So we are always looking to explore further opportunities and develop and advancing research in various therapeutic areas. So yes, there's definitely an interest in that, but there is, as of now, no compound that moved into the clinical stage so far.
Okay, thank you. I think we're going to now turn to the panel questions, right, Eric?
Yeah, so we've had some questions coming in from some of the families watching at home, so we're just going to kind of shoot those around to the panel. And the first one I'm going to direct at both Diane and Leslie is, we of course have interest from families about other exons that might be, in the pipeline. We obviously have had a lot of companies with focus on 51, 45, 53, some of the ones that have a kind of a larger share within the Duchenne population. What are the plans with your companies in terms of pipeline of these other exon skipping targets? And a follow-on to that as well is we've seen with Sarepta and the Essence study where multiple exon skipping investigational products were kind of put under the umbrella in one trial. Are we looking at more trials like that where we can kind of move this along at a little bit of a faster pace rather than development of one at a time? And I'll start with you, Diane, and then we'll go to Leslie.
Sure. Another great question, Eric. So, I'll start off by saying Sarepta, we are 100% committed to developing medicines for 100% of the individuals living with Duchenne, and of which I believe up to 85% could benefit possibly from exon skipping. And so we definitely do have plans to expand into other exons. Like I said, we are looking at our data. We have to look at both our PMO platform and Ryan referenced next generation. So we have our PPMO platform. And so we really will have to make a decision with respect to which platform to carry forward. And so we certainly, as we develop those plans, will be communicating with the community. I do want to make reference because this community has been out in front with respect to looking for a streamlined path for, the more rare mutations going back over a decade. I remember talk of a platform type of approval. And so I think a lot of progress has been made over the years, particularly I referenced legislation, 21st Century Cures included a provision in there that really codified a regulatory foundation to be able to leverage data in the situation where you're using a genetically targeted platform, across different candidates. And so, I think the regulatory foundation is there to develop a streamlined pathway for the more rare exons. I think, our data and the science needs to catch up. I think FDA is certainly open. And I think it really is incumbent upon us, the sponsors, to think creatively and do things like the master protocol trials and basket trials and things like that as we gain more evidence around our products. So I do have high hopes that we'll be able to go faster after these initial set of exons.
Fantastic. And Leslie, obviously you shared, exon 44 is coming. Does NS Pharma have an idea of kind of what the next targets would be after that?
We're considering another compound. We do have a library, a portfolio of compounds. So we are trying to make internal decisions about what would best fulfill unmet medical needs, what would be the best compound to then bring forward. But we expect to shorten the timelines between the rollout of these next compounds. So we're taking our learnings from the first compound that we took through the approval process, Ataluren, Viltepso, and we're applying that to exon 44 skipping and then subsequent compounds as they come up.
Okay, great. And as just a quick follow on that, this might be a little bit early, but are you planning on maintaining that same 80 milligram dose that you had with Viltepso for your other products like Ataluren, or is that something you're still working on?
So it's a little too early to speak to that. We are looking at a variety of doses so that compound is already in the clinical trials in Japan. That's on clinicaltrials.gov. And it's in clinical trials in Japan. We're going to wait and see what the results from that look like in order to help guide us and steer us into the appropriate dosage for our US studies of the phase 2 program for that exon 44 skipping agent.
Okay, great. And Christian, of course, one of the questions that we've seen a couple of people comment with is, can you expand on kind of the regulatory filing status for Ataluren?
Sure, absolutely. So as I said in the presentation, Ataluren is so far an investigational medicinal product in the US still. And we are in a continued discussion with the FDA on potential approval pathways. So more to come on this one.
Okay. And this one is for Diane. With regards to the Mission DMD study, could you expand a little bit on what those the high doses are that you're looking at in that trial?
Yeah, I don't believe we have spoken to the exact dose levels. It is significantly higher than the approved 30 mg per kg dose, and to unprecedented levels. So, yeah.
Okay, thank you.
Great. And we just want to thank all the panelists. We had a lot of questions come in. We are going to submit those to all the sponsors after the conference. And we appreciate your time today. We're going to now turn to introduce our first Pat Chat session. Pat's going to be talking with Dr. Eddie Smith. So Pat, handing it over to you now.
Thanks, Ryan. Thanks, Eric. I'm looking forward to hearing from Dr. Smith, as I think all of you are. Dr. Smith joins us from Duke University, where he's running multiple trials to include gene therapy trials. Dr. Smith, thank you for joining us. How are you this evening?
I'm doing great, Pat.
Good.
Thanks for having me.
Good. We so appreciate your time. So I don't know if you were able to listen in, but we've just talked about dystrophin expressing therapies. And I'm going to back up before I ask you my question about that. But I think that, in the 2018 care considerations, what we saw is the mean age of death changed and added 10 years of life to these young boys. And that was employing known medical tools, steroids, steroid regimen, cardiac medicines, pulmonary care, physical therapy, et cetera. So with these dystrophin expressing compounds, and certainly with the gene therapies, are you seeing the curve change as well? Are you seeing, as you see these patients, that things are once again changing toward better trajectory of longer ambulation, better pulmonary function tests for longer periods of time?
Wow. Well, Pat, my honest answer is I don't know at this point. I still think it's a bit early, and you're right. I mean, we've made strides with improved care and improved cardiopulmonary care in the absence of any dystrophin replacement. But of course, this is the big question. If we can begin to help our boys and young men make some form of dystrophin, some functional form of dystrophin in whatever amount, the hope would be that we would shift things even further over to the milder side, and then improve quality of life, improve function, or at least help maintain function even longer. There are a lot of factors that will figure into how effective that approach is. How much of the dystrophin does the treatment allow the boy to make? At what stage of Duchenne are you intervening? What's the condition of the muscle at the point when you introduce these treatments? And then also, very importantly, what is the quality of the dystrophin that a particular treatment allows a patient to make? We don't really have, when we talk about gene replacement therapy or exon skipping, it's really important to remember that these are not normal full-length dystrophin proteins that are being made. They're shortened versions, and they're going to be less effective than full-length dystrophin will be. And again, it's being introduced at some point well into the disease course. So I'm kind of jumping ahead. I don't think I answered your question very well, but these are the things that I think about: how much dystrophin, and what's the quality of that dystrophin, and what's the capacity of the muscle at that point in time when you introduce this treatment to respond to dystrophin, for that dystrophin to have something to latch onto and have a functional impact.
Yeah, I think it's so hard, right? And I think one of the other questions that parents are asking, in fact, just the other day I had a mom call who said, I have a young boy, gene therapy studies are underway. He's also amenable to exon 51 skipping. So how do I think about that, right? And how do you think about that in terms of, can you layer these therapies on? What would you need to know in order to think gene therapy and then an exon skipping product after that? What would you need to know as a clinician to be able to prescribe something like that?
Well, first and foremost is always safety, right? And I just based on what we know right now, I don't know why there would necessarily be a considerable safety concern by combining those two therapeutic approaches. In theory, they should work synergistically as well, and they're not going to combat each other. One will make mini or micro dystrophin, and the other will help make skipped 51 skip dystrophin. One question I have, and I think many people have, is back to this quality question: is what's the quality of a skipped dystrophin? We know it's a much longer dystrophin protein than the mini or micro dystrophin that's being produced through gene replacement therapy. But those gene replacement therapy constructs were based on patients out in the world who had very short dystrophin molecules, but really relatively mild Becker muscular dystrophy. So the size of the protein is not the only thing, but it may be important. So I don't know the answer to that, Pat. I think we'll learn as we go. But at this point, I don't see a reason why they would be somehow mutually exclusive. One big hurdle, oftentimes, if you find the safety acceptable and think there might be a reasonable chance for additional benefit from combined therapy, is then getting it covered by a payer, right? And convincing, say, and I'm talking outside of a trial, convincing a payer to pay for the two treatments together, because as we all know, these are not cheap therapies.
Yeah, I think we're all struggling with that, right?
Yeah.
I think that, obviously if you have one or the other therapy first or participate in a gene therapy, should there be approval, what are payers going to do to look at that therapy that will be incredibly expensive? Although from the parent side of things, getting dystrophin expressed is sort of like the holy grail, right? And then thinking about a gene therapy with a microdystrophin to add to or supplement or synergize with that dystrophin protein would be pretty incredible opportunity. Right. At least I think we'd all think that and hope it would really change what's happening in terms of progression. So we're working on newborn screening. I mean, I'm very hopeful there'll be a day when we can think about a baby and diagnosing very early and then intervening. I also think that, I worry a lot about the language we use, change Duchenne to Becker, because I think we're changing Duchenne, right? We want to get those three flavors of outcomes that you talk about: this slowing progression, stopping progression, or providing benefit. So would you treat a baby with a weekly antisense oligonucleotide?
I would. I think that we have a lot of progress yet to make with the ASOs, and there are so many promising and interesting programs in the pipeline that really seem to have potential to induce more exon skipping and then produce more dystrophin and then hopefully have more effect. My current thought is that that as far as dystrophin replacement strategies go, and this may end up being incorrect, but I'm more optimistic in the short term with gene replacement therapy approach in terms of amount of dystrophin, importantly, amount of mini or micro dystrophin. That's one thing. It's one thing I always try to bring up. We talk about dystrophin replacement, but it's very important to remember that these are not normal dystrophin molecules. Mm-hmm. And to your point about Becker, it's well taken. We're learning to use this term dystrophinopathy, I think, more and more. In those terms, I guess what we would be talking about is shifting towards a milder form of dystrophinopathy. And the quality of the product and the quality of dystrophin that's made is key to that, but also, as you just mentioned, the age at which therapy is introduced. So I would not rule out prescribing exon skipping therapy to a patient. And if a family came to me and was very strongly pursuing that, I would not say no. I think many providers would not say no. But getting that approved, getting it covered, and it is potentially another story. I would also say that you mentioned a while back about exon skipping and in the era of gene therapy trials, and so for that family who has a boy who's exon 51 skip amenable, but also looking hard at trying to get into a gene replacement trial. Being on the exon skipping therapy is not necessarily exclusive. There were just, I think, the programs I'm aware of do allow a washout period. So the fact that you started or were on an exon skipping drug, but your child would have to come off of that to be eligible for the current slate of trials that are available or hopefully soon to be available.
But one day, hopefully, combination therapies, right, where we can treat with exon skipping or a microdystrophin and then lead right back into the next gene expression the next dystrophin expression therapy.
Well, I think it's right. And I think that there's some analogies here to the spinal muscular atrophy world. These are none of the things we're talking about are cures. I think we can talk about approaching that as we intervene earlier with more effective therapies, but there's always going to be that need and that desire to do more, and on top of the current standard of care. So I don't see any way around in the near future. I don't see any way around combining therapies. Again, not to harp on this, but I think the key at the end of all of this is going to be to convince payers to pay for that, and they will want to see evidence that additional treatment provides additional benefit on top of the baseline therapy.
Right. Well, we have to convince payers. We have to do the work that we need to do to see it, and then convince payers. I'm so grateful for all you do. Thank you for spending time for us after probably a long and busy day. So we hope to see you next year in person. Thanks.
Likewise. Yeah. Thank you, Pat. Good to see you.
Good to see you as well. Thank you again.
Bye-bye.
Goodbye.
Bye.